Not to sound redundant, but yes, I am Becca. I was the Lee Fellow in Evolution and Molecular Biology, and this summer I completed my fellowship at North Carolina State University in Raleigh, North Carolina. So this summer I was at the North Carolina State University Genome Research Laboratory, specifically in the lab of Dr. Brian Wiegman, who is the Executive Director of the Genome Research Laboratory. Dr. Wiegman also assumed the role of my site mentor for the duration of my fellowship. His picture is there on the top. I also spent a lot of time in the molecular lab with our lab technician, Brian Castle, who's in the middle, and I shared an office space with their current PhD student, Keith Bayless. Together, the four of us comprised the Wiegman lab in its entirety for the duration of the summer. So we were a very small lab. We all got to know each other very well. Clearly, I was the only undergraduate intern in the Wiegman lab, and I was actually the only undergraduate intern to be hosted in the Wiegman lab in quite a few years. So I'm very proud of that fact. So the Wiegman Lab is known internationally for its work on the Fly Tree Project. The Fly Tree Project is an international research collaboration to elaborate the details of fly diversity with the ultimate goal of providing a newly resolved phylogeny for the fly tree of life. And Dr. Wiegman and his collaborators have been able to make great strides on such a massive undertaking because they have access to an invaluable sequencing technique that allows them to get immense volumes of raw DNA sequence data, and that technique is referred to as anchored hybrid enrichment, which is able to sequence over 500 genes in 48 widely divergent model and non-model taxa. So my goal over this summer, my fellowship was an independent research project to investigate the evolutionary history of Blephronura, which is a genus of neotropical South American fruit flies in the Tephrididae family. My work is really near and dear to the heart to one of our professors of biology here on campus, Marty Condon, who has identified over 40 new species of these fruit flies and described incredibly amazing and very specific interactions of these fruit flies with their host plants and especially with their parasitic wasps. So flies in the genus Blephronura feed on plants in the family Cucurbitaceae, also referred to as cucurbits. So cucurbit flowers then are host to many species of Blephronura that feed on and lay their eggs specifically within very particular sex flower tissues of single species of cucurbit plants. Bolopius wasps, these are parasitic wasps. They represent an important third trophic factor in Blephronura communities. These parasitic wasps lay their eggs inside the developing egg or larvae of the Blephronura flies. The fly is not killed immediately, however. Rather, the fly continues to feed on the host plant tissue, while the parasitic wasp feeds on the tissue of the developing fly. Until ultimately the fly dies before it's able to reach adulthood. Instead, then, when the parasitic wasp reaches adulthood, it is the one that emerges from the plant. One of the very interesting things about this system, this is a typical predator-prey system that you would see on the Discovery Channel, but with a new twist. So these Bolopius wasps are so incredibly specific as to the specific species of Blephronura they are capable of infecting, that if the parasitic wasp infects the wrong fly, the wasp egg dies. So my role in investigating such a multifarious system was to focus on the evolution of immune system genes that may contribute to why one fly is able to escape parasitism and another one to be succumbed by it. So it was the role of Dr. Wiegman, Marty Condon, and I to first establish a database of immune system genes that, when sequenced through anchored hybrid enrichment, would reveal known parasite-host relationships and could be used to test genetic and molecular mechanisms that govern host survival. However, unfortunately, little is known about the immune system genes of Blephronura. However, I was able to draw comparisons from its close relatives, including Drosophila, the common fruit fly that frequents many high school biology classes and that you see flying around your bananas or your garbage. Also, other close relatives in the Tephrididae family, including Ceratidis capitata and several species of Bactrocera, among others. So I started by researching scientific literature, and I was able to identify 144 immune system genes that are known to be associated with the cellular immune response in Drosophila against Drosophilus parasitic wasps. So this was really the key foundation of my research. Next, I went to protein databases online, especially through OrthoDB. It's a very scientific website. And I looked for these specific Drosophila genes and tried to find them in other Tephrididae genomes, specifically in Ceratitis capitata. And out of the 144 Drosophila genes, 81 could be found in Ceratitis. From there, I sought to narrow down our database selection. I did this first by looking for genes of high similarity. And I accomplished this by building protein alignments, which is just a very scientific way of saying a supermatrix file that is used to represent gene similarity. Furthermore, I was able to use these databases to find single copy orthologs. So when we're talking about building trees, single copy genes represent the best way to do that. And so I further narrowed down our database by eliminating those with multiple copies in the genomes. And so out of the 81, 56 exist as single copy genes in multiple tephritid species. As the final icing on the cake, I then built nucleotide alignments. Nucleotide alignments, just like protein alignments, are used to determine gene similarity. However, these nucleotide alignments will be absolutely crucial to the next part of this research project, which will be ultimately probe design. So in order to sequence through anchored hyperenrichment, we have to have specially designed probes that go in, find our target genes, cut them out so that they can be sequenced. And luckily for us, out of the current anchored hybrid enrichment kit that's used in the Wiegmann lab, 4 of these genes of interest are already included. So less work for us. So this summer was a once-in-a-lifetime research opportunity to see the innermost workings of one of the pioneer labs in the country for phylogenetic analysis and evolutionary biology. I was able to shadow a professional lab technician, although he was very tickled when I called him a professional. He is, I assure you. And I was able to learn molecular techniques that are left out of most laboratory textbooks and instruction. I was also able to build an independent research project under the attentive help and supervision of Dr. Brian Wiegmann, a world-famous entomologist and evolutionary biologist. As the only undergraduate intern in the Wiegmann lab, I essentially lived the life of a graduate student. My schedule was my own to rule. My project was my own to construct, troubleshoot, and see through. I was also able to connect with scientists and researchers from across the country and around the world. Cat sitting is actually a form of scientific networking, believe it or not. But the process of scientific collaboration incorporates more than just the sharing of scientific information. It's also the sharing of scientific experiences. And so through the lives of my new colleagues, this summer was a colorful and insightful learning experience, just beyond words. And I was also able to discuss my career options, seriously analyze them, and discuss graduate school opportunities with individuals from across the country, around the world, North Carolina, South Carolina, Georgia, Brazil, and even Australia. And of course, it was just great fun. So I got to see the Atlantic Ocean for the first time. I went cliff jumping for the first time. This is not me. I did jump, though, 3 times. I jumped 3 times. And I was able to experience the 4th of July in downtown Raleigh, where they have a wonderful fireworks show. And of course, I could not have completed this without the financial aid and support of the Cornell Fellows Program. I would like to specifically thank my site mentor, Dr. Brian Wiegmann, who unfortunately couldn't be here. Last I heard from him, he was in Germany. At a Diptera conference, of course. It's all about flies. I would also like to thank my faculty sponsor, Marty Condon, and my academic advisor, Craig Tepper. And of course, I would like to thank my family and friends who have supported me throughout this experience and who continue to support me today. Thank you.